Okay. So hi, everyone. Welcome to this seminar. So it's co-organized by the French speaking theoretical biology society and the applied math of Le Havre Normandy University. And it's our pleasure today to welcome Megan Morisson, who is a postdoc right now at Courant Institute, New York University. And she will talk about the nonlinear control in the nematode C. elegans. Thank you, Megan.
Hi, thank you so much for this opportunity to talk to you guys today. So I'm going to be talking about stuff that I did during my PhD, nonlinear control in the nematode C. elegans, as well as some previous C. elegans research. This is stuff that I did with Nathan. Oh, oops, I was muted. Okay. I guess I just said thanks for the opportunity to talk today. I'm going to be talking about nonlinear control in the nematode C. elegans. This is work I did during my PhD with my advisor Nathan Kutz in the applied math department at the University of Washington, and then also Charles Feisler, who's now a postdoc at the Research Institute of Molecular Pathology, the University of Vienna, and he does C. elegans research there. Manuel Zimmer. So first I'm going to give a general overview of C. elegans. I'm going to talk about modeling their behavior with Markov models. Then I'm going to talk about representing the neural dynamics of the neurons in PCA space, linear models for these dynamics, and then global nonlinear models can be used. So C. elegans are a great model organism for neuroscience because they don't have that many neurons or behaviors. Their neurons can be divided into three groups: sensory neurons that sense a variety of things such as pheromones, odors, temperature, etc. Then that information gets passed on to interneurons to kind of integrate that information, proprioceptive feedback, and then output information to the motor neurons that control the body bends and swimming behavior of C. elegans. So this is a video showing calcium imaging of the neurons in the C. elegans, so the interneurons. And so since there aren't that many neurons, this imaging, they can sort of pick out the activity of the different neurons and label them. The C. elegans, they also don't have that many behaviors, so they swim forward, they swim backwards, they do these various turns. So their very simple nervous system only has 302 neurons. They have a stereotyped connectome, so they're connected in the same way across animals. And then a lot of those neurons are named and implicated in different behaviors. And then they have only a few behaviors, and so they're a great organism to model sort of neural dynamics, integration information, and then how that produces behavior. Here's a couple more videos of forward swimming. The black dot is used to track the location and the speed over time. And then here's sort of a reversal, and then it has this like pause state that it also displays. So the main categories of behavior are forward, reversal, there's dorsal turns, ventral turns, pause, and then this quiescent state that kind of corresponds to sleep. So here are some previous C. elegans models. There's this Markov model that modeled clusters and transitions between different macro scale behaviors. So this isn't looking at neural dynamics, this is just looking at the behavior and how the C. elegans transitions between behaviors over time or given different environments. Then there's linear switching, which looks at the local linear dynamics of the neural activity. And then this model makes a connection between the behaviors and then the dynamics. There's linear controls, which disambiguates the control mechanisms in the neural dynamics and then the more intrinsic dynamics. And then what I work on, which is nonlinear controlled, which is this global nonlinear model instead of having a patchwork of different linear models. It's just a single model. So first, I'm going to talk about stochastic switching between different behaviors. This paper looks at the geometry of locomotive behavior in C. elegans. The main reason why C. elegans swim around is to find food. So there's this roaming behavior, it's an exploratory behavior, and dwelling, which is an exploitative behavior. So worms, they have to decide whether or not to kind of eat the food in the food patch they're currently in, or maybe swim around, move on, try and find a better food patch. And then once they've eaten a lot, they will enter this quiescence state once they're satiated. And these researchers found that locomotive activity and the probability of switching between these different behaviors is determined by nutritional status. So they built the stochastic switching models between these three states, which is roaming, dwelling, and quiescence. And they found that given like good food, high quality food patches, the probability of transitioning into the green dwelling state was very high, and it tended to stay there. Given lower and lower quality food patches, however, the probability of transitioning into this roaming state increased quite a bit. So by varying the environment of the C. elegans, they could change these transition probabilities. And so they created this model and then kind of analyzed how this behavior sort of maximized the C. elegans' ability to survive by finding high quality food and exploiting the food that it did find. A little bit more detailed model. I was looking at a stochastic neuronal model it predicts random search behaviors at multiple spatial scales in C. elegans. So there's these two groups of neurons in the interneuron class. One is responsible for forward swimming behavior, the other is responsible for reversals. And these two groups, they're attached to one another and they inhibit each other. They get information from sensory neurons and interneurons and then output to forward motor neurons and then reversal motor neurons. So this stochastic switching model models the forward neurons as sort of one unit, the reversal neurons as another unit. And there's some probability of them switching on or off and inhibiting one another. So they use these units to create a Markov model representation of the command neural network, and then simulated the dynamics that would happen with this model, and then compared the simulated dynamics to sort of neuron behavior and C. elegans behavior from experimental data collection. So the next level of model connects calcium imaging neural dynamics to the behavior that's displayed. And so what the Zimmer lab initially did is they found that global brain dynamics embed the motor command sequence of C. elegans. And so they measured the behavior over time, but they also measured the activity from all these neurons. And so here, this is a low-dimensional PCA representation of the neural activity, and then the color is the behavioral state. And so they found the different regions of neural activity in PCA space corresponded to different behaviors. So they broke the behavior down into these major groups: forward, slow swimming, reversal, ventral, and dorsal turns, and they mapped out what transitions happened from each behavior to each other behavior. So after a reversal run, for example, the C. elegans tended to transition either into a ventral turn or a dorsal turn. After a dorsal turn, it would then transition into swimming, slow, or fast. They also then made a connection to the neural activity. So this is a picture of calcium imaging. And then they assigned labels to each of the neurons. This is a difficult process. Sometimes they're not able to disambiguate between different neurons. And they're also sometimes not able to get a good measurement of the voltage for each neuron. So this is definitely an active area of research in the experimental community, getting really good, high quality imaging of the neurons and then being able to label them all. So once they had this data, then they performed PCA on the neurons. And this that allowed them to look at the activity in a low dimensional space instead of looking at all 100 neurons. And so they have this data matrix X where the columns, these are the different neurons, or the rows are the neurons, and then the columns is the calcium imaging over time.
Sorry, Megan. Do you know what's the order? Because it looks like they are grouped. The red are with the red, and so what's the order? It's by distance, or it's?
Yeah, so they sorted this data by the major activity, the behavior that was most associated with each neuron. So for example, this first cluster, and I'll show a more detailed picture later on, these neurons are most highly associated with like reversal swimming, and the next one. Yeah, and so that's why you're able to see these distinct patterns, is because they did this clustering. So SVD and PCA, they're very related. When you do SVD on the data, you get these less singular vectors, which are PCA modes. They're orthogonal. You have your singular values, which tells you kind of how important each mode is, how much variance each mode describes in the data. And then you have your right singular vectors. So this is the time series of each of those PCA modes or SVD modes. If you do PCA, you get similar information. You get these right singular vectors, which gives you the time series of the PCA modes, and that's similar to what you get out of SVD. So what they did is they plotted the time series of the principal components over time. And so this is showing the time series in PCA space, W1, 2, and 3, and then the color is the behavioral state. So when this elegant is in this preferred low oxygen state, it tends to be in quiescence. And so in PCA space, the activity clusters here. And then when it's in this adversive high oxygen state, it goes through these behavioral transitions of a reversal, then a turn, and then forward. So it transitions a little bit more active. So this is looking at now the neurons, the names of the different neurons and what's in each of these PCA modes. So each mode, PCA mode 1, 2, and 3, has a loading for each neuron. And these loadings are kind of different dominant patterns that show up in the data. So neurons that have kind of highly correlated activity or anti-correlated activity with others, they may show up like very positive or negative in different PCA modes. So for example, these first neurons here, the different colors correspond to different behaviors that they're associated with. And so these neurons are, they have very correlated activity, and they're anti-correlated with these other pink neurons. So when the PCA mode is in the time series, when it's very high, when it's on, that means that these neurons are very active, and then these are the opposite, they're not active. And then vice versa, when PCA mode is very negative, that would mean that the pink ones are active and then the red ones are inactive. And so this gives us an idea of how these neurons kind of cluster which ones are correlated or not correlated for different patterns. All right, so those are the neurons. Now we can look at the time series associated with each of these modes. This is the time series of the first 3 PCA modes. And as you add more PCA modes, the percentage of the variance that you're able to describe goes up, right? So if you include 2, then 3 PCA modes, you're able to explain 60 to 70% of the data. And the idea is that we can just look at the first principal components, the time series of these first modes, and then get a good idea of what's happening. We are throwing away some of the data, some of the variance, but it's not as relevant as sort of the dominant activity. So now I'm going to be talking about dynamical systems models for these neural dynamics. So far, I've talked about, okay, what does it visually look like in PCA space, but can we fit a dynamical systems model to this? So in Linderman et al., they created this hierarchical recurrent state space model. They found it reveals discrete continuous dynamics, the neural activity of C. elegans. So what they did is that they took this neuron data and then they sort of cut out the dynamics that corresponded to different behaviors. So everywhere you see yellow, that's one sort of behavior that's displayed. And so they cut out the dynamics for that yellow behavior and then fit a linear model for that. Similar with green, they cut out the dynamics and fit a linear model. And so this is what the dynamics of the different linear models look like for the different behaviors. In A, they kind of patch them all together, and B is showing the different individual linear models, so it's piecewise. And then they also have a probability of transitioning between the different models. Linear models, they only have one fixed point. You need some sort of transition between them to get the full dynamics. So they fit models a couple different ways. They fit a model for each worm, and then they fit a model in a non-hierarchical way and then in a hierarchical way. So in the non-hierarchical way, they just used the behavioral state to inform the probability of transitioning. Versus in the hierarchical model, they also used the state space to inform the probability of transitioning. And this gave them a much better model when they used the behavioral state coupled with the sort of neuron state to determine the probability of transitioning. All right, so next I'm going to be talking about combining multiple linear models into a single nonlinear model. So can we build a nonlinear model for the latent variable dynamics? This is what I worked on during my PhD and something I'm still working on. So here are the time series of the first 5 principal components over time. The idea is instead of creating a patchwork of linear models, can we create a single nonlinear model? So here's example calcium imaging time series, and then the principal component analysis just showing the first 2 PCA mode time series over time. So in the Zimmer model, they use 3 PCA modes here and just looking at the first 2, and then they're colored by their behavioral state. So I fit a simple dynamical systems model. I'm going to show the deterministic version of the system, but because the data is noisy, there is stochasticity. We used a stochastic version of the model where there's this noise term tacked on. And this was also an important part of the model, allowing the dynamics to transition away from different fixed points. So what this, the simplified version of the model has is it has 2 stable states at plus and minus 1, a saddle fixed point in the middle, a damping term, and then a control pulse that's able to turn on or off during transitions. And so this general form fits the data because the forward and backward swimming states kind of correspond to stable attractors, and then they transition between these with this state in the middle that is unstable, where sort of a decision is made. Is it going to transition to the forward swimming or the reversal state? So different parameters have different effects on the model. For example, changing beta affects the relative stability of the two fixed points. So as you increase beta, that would decrease the stability of this right fixed point. And then the dynamics can more easily shift away from that fixed point. And so with stochasticity, you could transition over to the fixed point, but because the noise in the system and the stability region is small, it can easily transition back over to that left fixed point. Another way to get a transition is with a control pulse that also kind of moves the system through bifurcation, and then you can transition from the left to the right attractor or vice versa. All right, so I fit this model to the different Elegans data. There were 5 different data sets by fitting the first PCA mode over time to the X variable, and then allowed the control signal pulse to turn on when the behavioral state was showing a transition. So here's an example of a fit model to the Elegans data. You can see that the forward swimming is clustered over here. The backwards swimming is also clustered in the right state. And then you have transitions between the two. So this is a very simplified and constrained model. There aren't a whole lot of parameters. It's minimally parameterized. But the idea was to get sort of the main transitions and behaviors without having too many parameters to avoid overfitting. So with a nonlinear model, we can see that there are multiple mechanisms that are necessary to support each behavior, and then perhaps also similar mechanisms that are underlying what may seem to be different behaviors. So for example, the Elegans, they have these reversal bouts where they're in this reversal state, they'll transition into a forward swimming state, but then quickly transition back to a reversal state. And this type of behavior can be induced by changing the oxygen levels. So if you increase the oxygen level in its environment, this forward swimming state becomes kind of destabilized. It won't spend a whole lot of time swimming forward. And so how this corresponds to the nonlinear model is it shows maybe there are perhaps 2 distinct mechanisms that are responsible for supporting forward swimming. So for example, you need to have that kick or transition to a forward swimming state, but the forward swimming state also needs to be stable. You have to have a stable attractor in the neural dynamics. And if it's not stable enough, then the dynamics will transition back to the backward swimming state. So this may show that there are different mechanisms that are responsible for the transition to the forward swimming state, but then also stabilizing the swimming state. And if we can sort of disambiguate what the different mechanisms are in Elegans and what's responsible for those different components, that could be interesting for understanding how Elegans sort of makes decisions or is able to change its behavior with different neuromodulators. So in summary, Elegans behavior can be described using Markov models, neural dynamics. They can be modeled in this low dimensional PCA space. So you don't have to look at all 100 neurons, you can just look at the principal components and kind of understand what's happening in the dynamics. Different regions of the latent variable space correspond to different behaviors, experimentalists have found. And then the latent variable dynamics, they can be modeled with either multiple linear models or a single nonlinear model. So once again, this is work I did with Nathan Kutz, Charles Feisler, and then Manuel Zimmer collected the data that we used in this. Thank you. Are there any questions?
Thank you, Megan, for this nice talk. Yes, there's some questions. So maybe we will stop the recording for the questions so we can discuss freely. And
Okay.